STEM to Schools conference. We want to thank all parents, friends, siblings, and teachers for coming tonight to support the STEM community and learn from three very interesting professional speakers about their respective fields. We also would like to thank Robin Cunningham, Dr. Siva Vidyanandan, and Dr. Robin Clayman who are all taking their time tonight to present their complex professions to a broader audience and inform and excite students about possible career paths in STEM. We would also like to thank Ms. Sabelli, our club advisor, for helping facilitate our organization's success for the past 2 years. Lastly, we would like to thank all our members of our club for their efforts in research, for not only contributing articles to our annual STEM journal, but also for the research that they are presenting tonight. We wouldn't be here today without each and every one of you. For those of you who are unfamiliar with the organization, STEM to Schools is a knowledge-sharing online platform whose goals are twofold. The first goal is to create an ecosystem whereby cutting-edge STEM research can be made accessible for high school students nationwide. The second purpose is to encourage collaboration among high school students interested in STEM to create greater awareness of STEM career paths. We accomplish these two goals by organizing conferences with leading scientific experts, publishing biannual STEM journals disseminating science articles from the perspectives of high school students, and conducting field trips to immerse our members into an array of STEM fields and applications in the workforce. For those of you who still have the opportunity to be a part of our club in the years to come, we are always welcoming new members with open arms. Our organization has grown rapidly since our founding nearly 2 years ago. Our hope is to spread the love of science, technology, engineering, and math across every school in America. We want to thank you all again for being part of this journey. Right now we're going to introduce our first speaker. We're going to have one of our members come up and introduce our first speaker.
Hi everyone, I'm Eric Sang from the STEM Schools Club, and I want to thank you all for coming to the 3rd annual STEM conference. I have the pleasure of introducing our first guest speaker of the night, Ms. Cunningham. She's a senior embryologist at Boston IVF, a cutting-edge institution that provides innovative fertility treatments in embryo science. Ms. Cunningham has more than 30 years of experience in human embryology and earned a Bachelor of Science in Medical Technology from Merrimack College. She is also certified as a Technical Supervisor in Embryology and Andrology by the American Board of Bioanalysis. Without further ado, please give a warm welcome to Ms. Cunningham.
Hello everyone, and thank you for inviting me to this very well-attended STEM meeting. I've done many presentations before at schools, and I've never had one with this many students, so kudos to you and your staff. I've been asked to present on embryology, which I have to say, of all the STEM fields, personally to me is the best, but I'm a little biased. I became an embryologist 30 years ago and never ever thought I would fall into the field. I was trained as a medical technologist and had my degree in medical technology. And I was kind of bored and had only been doing that for a couple of years. And I decided to look into getting into embryology. And at the time, embryology was a very brand new field. This was in 1989. There's only 1 or 2 clinics in the United States that were doing it, and very handful worldwide. And luckily, I was able to get a job with a company called IVF Australia, based in Port Chester, New York, actually originally based in Australia. And I trained as an embryologist there. Basically, it was done as in hands-on training because there were no embryologists at that time. Today, if you want to be an embryologist, you can get a master's degree in embryology from 2 or 3 universities in the United States. There are some abroad that you can get a master's degree in. And they will teach you a lot about embryology, mostly in animal models, and then you can come to a human clinic if you're lucky enough to get hired in one and become an embryologist. So that's pretty much the path that people would take today in order to get there. So I'm going to start off with the embryology laboratory. And we start with ovarian hyperstimulation. So what that means is normally a woman will ovulate 1 egg every month. And for those of us in the IVF field, that's pretty inefficient to have 1 egg. So we'd like to have a lot. So we will give the woman hormones and hyperstimulate her ovaries so we get between 10 and 12, hopefully, eggs. and then we will proceed with the egg retrieval. So up until that point, we're monitoring her ultrasounds and her estradiol and hormone levels. And when they are appropriate, we will give her a medication that will help to ripen and trigger those follicles and hopefully make the eggs be mature. And at that point, she will come into the laboratory, and we'll do an ultrasound on her. So this is an ultrasound, transvaginal ultrasound of the ovaries. And in ultrasound, fluid will show up, fluid-filled sacs will show up as black. So that's these sort of blistery circles here, and these are the two ovaries that are there. And the second picture is actually a cross-section of one of those follicles. So if you took it like a cantaloupe and chopped it down the middle, this would be the cross-section here, and you can actually see the egg growing inside the follicle of the ovary. So the patient's brought into the operating room, and the follicles are aspirated in the operating room. She is under anesthesia. It's not general. It's just regular IV sedation. Most patients don't remember it. The ovaries are punctured with a needle, and we aspirate out the fluid that's passed to a waiting embryologist. One of our embryologists right there, and that's actually our lab. She's going to scan through the fluid that's taken out and look for an egg. So this is what an egg looks like at time of retrieval. It actually is described as a cumulus mass, so that's this kind of gray fluffy area that's here, and the egg itself is right here. So think of it as if it were a chicken egg cracked on the counter. This cumulus mass would be the white of the egg, and that center dot would be the yolk of the egg. Usually at the same time as the eggs are being retrieved, the man is asked to produce a semen specimen. And there are many ways of processing that specimen, but what we need to do is to separate out the sperm cells from the seminal fluid and try to look for the cells that are the healthiest and the most motile or moving. So this all happens on the day of egg retrieval. So what we're doing is we're doing an insemination. So we're doing an insemination of either what we call conventional or ICSI. So with conventional insemination, we take the cumulus mass that I showed you before, put it into a labeled drop of culture media, and we add about 100,000 of the prepared washed sperm cells to each of the eggs. Then they're put into the incubator overnight, and we wait fertilization. The other way we can do fertilization is called ICSI. So it stands for intracytoplasmic sperm injection. So we do this under a micromanipulator. So those of you that play video games, and I'm sure there's one or two in the room that do, you would probably be really good at doing micromanipulation because we actually use joysticks and controllers in order to control the tiny microscopic tools that we use to inject the egg. So this is an example of intracytoplasmic sperm injection. I'll just give you a little graphic of what we have. On the left side is a holding pipette. It's just a large pipette that keeps the egg from rolling around. The egg is actually in the center here. And this is an injection needle, so it's beveled at the end, and there's a single sperm inside the tip of that injection needle. We're going to go into the single egg, inject it with a single sperm, and remove that needle. And at that point, the egg will close up its little hole, and we put it back into the incubator and await the fertilization check the next day. So this is what we're looking for the next day. And I say this in every talk that I give. This is my favorite picture in the entire world, other than my kids. But the reason I say that is that this is what we call a 2PN, or a 2 pronuclei, or a zygote pre-embryo. Has many names. In science, there's 12 names for one thing every time. What this is actually a fertilized egg at its earliest point. And this is called 2 pronuclei because it has 2 little circles in the center that represents the DNA, one from the egg and one from the sperm. So the cool thing about this is this contains the entire DNA profile to make it a full human being. Everything that we have comes from this. Still pretty cool. So this would be the day after the egg retrieval. We look for the fertilization. We put the embryos back into the incubator and culture them out for another 4 or 5 days. And at this point, we're taking them out almost on a daily basis and assessing their growth. So this is a 2-cell, pretty easy to tell, and here's a 4-cell. Here's an 8-cell. It's a little harder to see because remember, embryos are 3-dimensional, so we have to focus up and down. I can't do that on a slide, but in the microscope, it's easy to do. So when we get to day 5, we have what's called a blastocyst. So this is an embryo that has started its differentiation process. So the blastocyst is differentiating into basically two parts. One is this center group of cells called the ICM, or the inner cell mass. That's the part that becomes the baby. The second are these cells on the outside, sort of these half-moon circles. These are called the trophectoderm. And the trophectoderm is what becomes the placenta. So it's important that the embryo differentiate at this point, because you need to have both parts in order to have an implantation and a successful pregnancy. The ICM obviously for the baby, the trophectoderm for the healthy placenta. So at this point, what we would do is, if the patient were having a fresh transfer, we would transfer typically one embryo, unless she's probably had multiple failed cycles previously. We typically will only transfer a single embryo. We load the embryo up into a catheter, which is just a very thin tube. This happens to be one of yours, I think. It is a dual lumen catheter, so it has an outer catheter that's rigid and an inner catheter that's flexible. It looks just like a piece of linguine. It's very floppy. We load the embryo up between two bubbles, and we're going to place it into the patient's endometrium. So we're going to slide the outer sheath in, put the catheter in here, and deposit the single embryo into the endometrium. At that point, the patient is discharged. She's given some medication, and she's asked to come back in approximately 2 weeks for a pregnancy test. Now, as I said before, we're hoping for 10 to 12 embryos. We're going to transfer the best one. What do we do with the other ones that are left over? If they meet criteria and they're developing normally, we can freeze them. So vitrification is the process by which we freeze embryos at the current time. And basically, anytime you're going to freeze a cell, the important thing to keep in mind is that you need to remove all of the water from the cell and replace it with something. Because if you think of when water freezes, and I always use the example of frost on your window, it forms little crystals. Crystals are very bad for cells, because if the crystals form within the cells, they will rupture the cells and the embryo will die. Therefore, we need to put the embryos through a solution that removes the water and replaces it with a cryoprotectant. It's kind of like an embryo antifreeze. It's a solution that we put them in that does not form crystals when it is frozen. The embryos are then plunged and stored in liquid nitrogen, where they can be kept technically indefinitely. So this is actually our embryo storage room at Boston IVF. We have tens of thousands of eggs and embryos in there. They are stored in these little tanks and some of the big tanks in the back, and for safety reasons, I'm sure you can imagine, they all have wireless transmitters on them with temperature gauges inside of them. So that at any point, 24/7, 365, I can dial into any of those and find out the exact temperature. If any of those vary outside of our very tight range, it'll automatically call one of us, and we can come in, find out what's going on with the tank for the safety of the embryos. I'm going to talk about one more technique. This is probably the most STEM part of the embryology process. And it's one of the newer techniques, and I say newer, we've probably been doing it for about 15 years now. And it's called embryo biopsy. So, we can do this with pre-implantation genetic screening or genetic diagnosis. And the difference between the two of them is genetic screening is typically looking at chromosomes. So we can actually take some cells off of the embryo and screen them to see what the chromosomal makeup of that embryo will be. Pre-implantation genetic diagnosis is when we have a couple who is a known carrier for a certain disease, a single gene defect disease, such as cystic fibrosis, Huntington's chorea, Duchenne's muscular dystrophy. Anything that we know the gene mutation for, we can actually do the biopsy and diagnose those embryos pre-implantation. So we don't have to wait for the woman to get pregnant and she does a amnio to find out if the baby's going to be affected or carrying that disease. We can actually do it in the embryos before we implant them into the uterus. So the way that we do that is we have that blastocyst that I explained to you before. We're actually able to remove a few of the cells from the trophectoderm. There's probably 120 or 150 cells in the trophectoderm. We remove about 3 to 5, maybe 7 of those cells. So we're able to laser them off. We place them into a PCR tube that's labeled, obviously, and we send it off to a genetics lab. The rest of the embryo is frozen. So about up to 2 weeks later, we're going to get a genetics report from that lab, and it's going to tell us exactly which embryos are affected, which embryos could be carriers of the disease, and which embryos are free of the disease if we're doing a diagnosis. just a screening, it'll tell us the chromosomal makeup of each of those embryos. So with PGT, we usually freeze the biopsied embryos until we have the genetics report back. The report indicates what's normal and abnormal, and normal embryos can be used in a thaw cycle or for transfer. Abnormal embryos are typically discarded, or we have some research projects of places in Harvard and MIT that are doing research on affected embryos. So I'm going to see if this works.
How's it going?
Is it going? Okay, so hold on, let me stop that for a second. I'll tell you what we're looking at here. Maybe I won't stop it. There we go. This is an embryo. As it goes through, it started off at the 2PN phase. So what we're looking at is the embryo developing in what we call an embryoscope. An embryoscope is an incubator that actually has a camera inside, and it takes serial pictures of the embryo as it's dividing. The number down in the corner is the age of the embryo. So now we're looking at about day 3. You'll see it's about 8 to 10 cells at this point. As it goes through, it's going to form what we call a morula, which is just a ball of cells. This is the stage right before it starts to differentiate, and then it will start to blastulate. So it'll have a cavity that you'll see forming in the center. And the embryo will start expanding at this point. And the ICM, or the inner cell mass that I described to you, is actually so big it's moving off the screen right here. That's the ICM. And this is the trophectoderm here. So this is a normally developing embryo. You just saw life from day 1 through day 6 right there, in about a minute and a half. That is all I have for my talk today. If anyone has any questions.
10 minutes Q&A session after all the speakers.
Okay, we're all set then. Thank you.
Finish.